Hi everyone. In today's video, I wanted to go through the different types of mutations that can occur in DNA transcription. So, just to start off, there are 5 main types of mutations that can occur. And these mutations are categorized based on what we call the reading frame of the particular DNA sequence. So what does the reading frame mean? So, the reading frame is essentially the frame in which the codon of the particular RNA strand is read. So, say we have a particular RNA strand which is AUG, TTA, UUA, that should be, because we don't have thymine in RNA strands. GCC and AGC. Now, the reading frame would be the particular number of nucleotides, which is 1 codon, which equals 3 nucleotides. And this is the frame at which the tRNA molecule, when making the amino acid chain, will read the RNA sequence. So as AUG codes for methionine, the tRNA molecule will read this AUG with the particular reading frame and add this amino acid. Then for UUA, it will add another amino acid. Then for GCC, it will add another amino acid. And then finally for AGC, it will add another amino acid. So the reading frame is always 1 codon long, and this means that it includes 3 nucleotide bases. So what are the different mutations that can occur? Well, the first mutation that can occur is called an insertion. Now, as the name suggests, it is the addition of 1 or more nucleotide bases into the RNA chain. So say we take the previous example once again of AUG, UUA, GCC, AGC. If we have an insertion mutation, then a nucleotide base, for example, might be added in this section, and say there's another U added. And therefore, this new combination will look like AUG, UUU, and then now, as the reading frame is always read 3 nucleotide bases at a time, this A will move to this codon. Therefore, it will be AGC, because these two will now move 1 position that way, this one will move 1 position that way, this one will move 1 position that way, and that'll be the case for all of them. So it'll be AUG, UUU, AGC, CAG, and finally we'll have C left over. So as you can see, because there's been an insertion of 1 nucleotide base, we have the shift in the reading frame being towards the left. Because now that the RNA sequence has moved to the right, our reading frame has actually moved 1 nucleotide to the left. Because by adding 1 nucleotide base always, say in this position, we're always shifting the RNA chain in this direction, which means that the reading frame will always shift in the opposite direction. The second type of mutation, which is quite similar to insertions but just the opposite, is called a deletion. Now, deletions are simply the removal of 1 or more nucleotide base. So, once again, with our nucleotide combination of AUG, UUA, GCC, and AGC, if we say, for example, remove this nucleotide base, then the new sequence will be AUG, UU, and now, similar to an insertion, this nucleotide base will move to this position, meaning that there is UUG. Then these two will move to this position, forming CCA, which is another codon that's formed. And then finally, we will just have G and C here. Now, unlike an insertion, a deletion mutation actually involves the shifting of the reading frame towards the right. This is because once we remove a particular nucleotide base, the reading frame is actually moved to the right because the RNA chain becomes shorter. You have to always remember that the direction in which the RNA chain gets shorter is always the opposite to the direction at which the reading frame moves. Now, the third type of mutation that can result is called a silent mutation. These mutations are quite unique, as in terms of nucleotide bases, the change in the third base of the codon of a particular codon does not actually affect the particular amino acid that is coded there. So say, for example, we have our original sequence, AUG, UA, GCC, and AGC. Say, for example, we change this particular nucleotide base to being a G instead of a C. Now, as this particular nucleotide base is as this particular codon does not depend on the third nucleotide base in order to code for the amino acid, there will be no particular change in the amino acid sequence that results from translation. So even though the nucleotide combination will now look like AUG, UUA, GCG, and AGC, these two codons will still code for the same amino acid. And for reference, the amino acid that is coded by both of these codons is alanine. The next type of mutation that can result is called a nonsense mutation. In a nonsense mutation, there is a stop codon that is prematurely put in a particular RNA sequence, meaning that the process of translation stops before it can code for the entire protein. So say, for example, we continue on with our original sequence, and we have a nonsense mutation where this particular nucleotide sequence is replaced with an A. This would mean that the nucleotide sequence now reads AUG UAA. And as you may know, UAA actually codes for a stop codon. Therefore, this particular nucleotide sequence will end here. And these two nucleotide bases that are present here will not be coded for. The last type of mutation that I'd like to talk about is called a missense mutation. A missense mutation is essentially a mutation that results from a substitution of a particular nucleotide base that results in the change in a particular amino acid. Now, this missense mutation can be, for example, a combination of many amino acid sequences. So, with our original sequence, if, let's say, for example, this particular amino acid is changed to being a C, then The new sequence would be AUG, UCA, GCC, and AGC. Now, as the second nucleotide base in this particular codon has been changed, as opposed to the third, which results in a silent mutation, the amino acid that this particular codon codes for will be different. This is because UUA codes for leucine, and UCA codes for serine. So in conclusion, these are the 5 types of mutations that can occur. They are all in reference to the reading frame, which is the 3 codon sequence in which all RNA chains are read. An insertion mutation results in the insertion or addition of one or more nucleotide bases to shift the reading frame to the left. A deletion is when a particular nucleotide base is removed to result in the shift in the reading frame to the right. A silent mutation is when an amino acid is substituted for another. However, this substitution occurs in the third nucleotide base, therefore not changing the amino acid in any way. A nonsense mutation is when there is a stop codon prematurely placed in a particular RNA sequence to stop any further production of a particular protein. And finally, a missense mutation is when a particular nucleotide base is added to an amino acid sequence that is not the third nucleotide base, which would then result in a silent mutation, but rather the first or the second. This results in essentially the substitution of a different amino acid in place of the other one. I hope you enjoyed this video. Thanks for watching.